This apple is a source of sugar, and living things use sugar so that they can do cellular respiration to make the molecule ATP. ATP is how living things get work done, how we do maintenance, how we do repair, how we reproduce. Cellular respiration is how living things make ATP. And if you look at a diagram like this and you feel, oh my goodness, that is so terrifying. How am I ever going to master it? How am I ever going to figure this stuff out? Then don't worry. This show is about teaching you about cellular respiration so that you can ace your next test or the AP Bio exam. On the show tonight, we're going to talk about cellular respiration. We're going to have several songs of the week. We'll have excerpts from my songs about glycolysis, the Krebs cycle, and the electron transport chain. We'll have a weekly quiz, and we'll have tips for learning the greatest subject of all, biology. I'm Glenn Wolkenfeld, also known as Mr. W. I'm a retired AP Bio teacher, and my mission is about helping you succeed in AP, IB, or college biology. One of the ways that I do that is through my website, learn-biology.com, and its associated app, Biomania. I'll tell you more about those later. I have an ultimate AP Bio checklist that you can use to study for your unit exams and for the AP Bio exam. You can download it at apbiosuccess.com/checklist. It's really going to help you out. Let's start with our lesson of the week. We'll start with ATP and cell energy. All living things require energy for maintenance, for repair, for movement, for reproduction. Almost all energy pathways start with photosynthesis. And in photosynthesis, carbon dioxide and water are brought into autotrophs, self-feeding organisms like plants that in a reaction driven by light, photosynthesis, combine those carbon dioxide and water molecules to create carbohydrate and oxygen. The carbohydrate makes up the mass of the plant, and that forms the basis of all food chains. Photosynthesis is what we call an endergonic reaction. An endergonic reaction requires energy and it reduces entropy. So in this graph of the reaction, A represents the reactants, B are the products. And you can see that the energy level of the products is higher than that of the reactants. C is that energy difference, and D is the activation energy required to make that reaction happen. Endergonic reactions include photosynthesis, in which carbon dioxide and water, driven by light energy, create glucose and oxygen. And notice that there are 7 molecules over here, 12 molecules over here. It's decreased entropy. And any dehydration synthesis will tend to be endergonic. When you take monomers, combine them into polymers, that takes energy. Cellular respiration, which is our topic for tonight, is an exergonic reaction. Exergonic reactions release energy and they increase entropy. A in this diagram, just like the one we saw before, is the energy of the reactants. B is the energy of the products. And notice that the reactants have more energy than the products do. C is that energy difference. D is the activation energy. Examples include combustion. If you were to burn paper or wood, the energy of the reactant, the paper or the wood, would be much higher than the exhaust products, which are CO2 and water. Cellular respiration, which will be our focus for tonight, is an exergonic reaction. Here's glucose, here's oxygen, and here's carbon dioxide and water, the waste products. The purpose of cellular respiration is to create energy in the form of ATP that the cell can use. Notice that there are 7 molecules on the left side and 12 on the right side. We've increased entropy. Most hydrolysis reactions where polymers are made into monomers are exergonic. A key process in life is what we call energy coupling. In energy coupling, exergonic reactions are linked to endergonic reactions. And the exergonic reaction provides the energy that drives the endergonic reaction forward. Exergonic nuclear fusion in the sun creates the light that powers photosynthesis. Exergonic cellular respiration powers ATP formation. The breakdown of ATP to ADP and phosphate powers muscle contraction. It powers active transport. It powers dehydration synthesis reactions. It powers cell division. It powers how life gets pretty much everything done. Let's talk about the structure and function of ATP. You have a 5 carbon sugar, ribose, the same sugar that's found in RNA. You have the nitrogenous base, adenine, and we have 3 phosphate groups, one over here, and one over here. The function of ATP, we've said before, it's used to power work within cells. Every cell makes its own ATP. There's an ATP ADP cycle in which processes like cellular respiration will enable the cell to take ADP and phosphate and make it into ATP, and that stores energy for work. This PI over here, that stands for inorganic phosphate. And when you just have a phosphate group not attached to a carbon, it's inorganic, hence P sub I. To release energy for work, cells will remove this terminal phosphate over here, and that creates ADP and P and liberates energy through which work can get done. Now that we understand how ATP works, we can take a look at the big picture of cellular respiration and its first phase, which is called glycolysis. Cellular respiration is a redox reaction. That's something from chemistry, an oxidation reduction reaction. And during cellular respiration, glucose, C6H12O6, is oxidized to CO2, and oxygen is reduced to water. So here's the oxidation, here's the reduction. The main thing that you need to know as an AP, IB, or college biology student is that oxidation means the loss of electrons and hydrogen atoms. Reduction means the gain of electrons and hydrogen. So over here, glucose is losing all of these hydrogens as it's getting oxidized into CO2. Along the pathway of cellular respiration, which involves many reactions, there are intermediate redox reactions that reduce mobile electron carriers. And as they are reduced, they get transformed into their reduced counterparts. NAD plus is reduced to NADH. And it is useful and completely correct to think of NAD plus as a spent rechargeable battery with no more energy in it to power processes. But then processes like cellular respiration will recharge it so that it can provide energy to power various processes. NADH has a counterpart, and that's FADH2. And during cellular respiration, FAD is reduced to become FADH2. Now, when you look back at this diagram, you might see something that you didn't notice before. There are all of these NADHs over here, over here, over here, and FADH2 appears as well. Those are mobile electron carriers. And again, they're like rechargeable batteries that will carry energy over to the electron transport chain where most of the ATP in cellular respiration is produced. Glucose and its byproducts. Here's glucose. Here's another one of its byproducts, pyruvic acid. Here's another one called acetyl-CoA. They're progressively oxidized as NAD plus and FAD are reduced to NADH and FADH2. And in the last phase of cellular respiration, oxidation of NADH and FADH2 powers ATP creation. That's the very biggest picture of cellular respiration. Cellular respiration can be broken down into 4 phases, the first of which is glycolysis. During glycolysis, which occurs over here, energy in glucose generates ATP and NADH. The end product is this 3-carbon molecule called pyruvate or pyruvic acid. That's glycolysis, the first phase of cellular respiration. After that, we have the link reaction. And the link reaction links glycolysis with the Krebs cycle. What happens during the link reaction is that pyruvic acid or pyruvate is brought into the mitochondria. This orange line over here represents the mitochondrial membrane, and it converts pyruvate into acetyl-CoA. That generates some NADH over here, and it releases 1 CO2. That's 1/3 of the CO2 that we exhale. In the Krebs cycle, we have the complete oxidation of acetyl-CoA. And as acetyl-CoA is oxidized, we produce 3 NADH, one FADH2 and one ATP. Those are the useful products, and two CO2s are released. In the last phase of cellular respiration, we have the electron transport chain. And during the electron transport chain, all of the NADH, which was produced in the first phase of cellular respiration, glycolysis, it was produced in the link reaction, it was produced in the Krebs cycle. All of that NADH and the FADH2 that was produced in the Krebs cycle are oxidized to create electron flow, which powers phosphorylation of ADP into ATP. And that's where we make most of the ATP in cellular respiration. Where in eukaryotic cells do all of these processes occur? Glycolysis occurs in the cytoplasm. The link reaction happens as pyruvic acid crosses the mitochondrial membranes. There are two of them, an inner one and an outer one, and enters into the mitochondrial matrix. The Krebs cycle occurs in the matrix, and the electron transport chain and oxidative phosphorylation occur along the inner mitochondrial membrane and the intermembrane space. We can now talk about glycolysis, the first phase of cellular respiration. Glycolysis is a reaction in which energy in glucose generates ATP and NADH. And the end product is the 3-carbon molecule pyruvate, also known as pyruvic acid. So this is glycolysis over here. Now we're going to look at its details. Glycolysis involves 3 phases: investment, cleavage, and energy harvest. In the investment phase, enzymes phosphorylate glucose. That's a big word. What does that mean? It means that they take a phosphate from ATP and jam it on to glucose. That creates the intermediate compound glucose 6-phosphate. And then enzymes repeat that activity by taking another phosphate from ATP, jamming it onto glucose 6-phosphate, rearranging it a bit. We wind up with this molecule, fructose 1,6-bisphosphate. That leads to the next phase, which is cleavage. A cleaver is a tool that's used to cut something in two. In cleavage, this intermediate compound, fructose 1,6-bisphosphate, is split into 2 G3Ps. That stands for glyceraldehyde 3-phosphate. The truth is that I'm simplifying a little bit, and one of these G3Ps is actually in a slightly different form, and there's an enzyme that converts it into G3P. So after cleavage and this slight modification, we wind up with 2 G3Ps. Note that every arrow in this diagram represents an enzyme, and there are 10 enzymes that are involved in taking glucose and processing it during glycolysis. The harvest phase, in parallel, this happens twice because we have 2 G3Ps. G3P is oxidized as NAD plus is reduced to NADH. So here we have NAD plus becoming reduced to NADH. Enzymes also phosphorylate 2 ADPs into 2 ATPs for each G3P. So here's our gross and net yield. The gross yield is 4 ATPs because 1, 2, 3, 4 ATPs are created over here. We create 1 NADH over here, 1 NADH over there for a total of 2, and 2 pyruvic acids result as the end product. But remember that 2 ATPs were used up in step 1, so the net yield of glycolysis is only 2 ATPs. You have to take these 4, subtract 2 NADHs, 1 over here, and 2 pyruvic acids. That's probably worth memorizing. I have a song about glycolysis, and we're going to play the intro up to the first chorus. Here we go. Hey, sugar. Come on over here, sweetie. Let's break it down. Glycolysis is a series of reactions, enzymatic actions, energy transactions. Takes glucose, a molecule so sugary, breaks it down to NADH and ATP. It's an anaerobic cytoplasmic pathway that amazes, organized easily into 3 phases: investment, cleavage, and energy harvest. Tell me later which one you like best. Investment, activation, energy supplied. Cleavage, our 6 carbon sugar divides. Harvest, we get our energy yield. So beautiful, so intricate, keep your eyes peeled. Glycolysis, come on sugar for the breakdown, for the breakdown. Investments, like striking a match, that energy you put in makes the fire. So that's glycolysis. And I want to let you know that if you're feeling like this material is complex and you need additional support, there are tutorials about all of this material on learn-biology.com. Tutorials about ATP, about cell energy, endergonic versus exergonic reactions, and about glycolysis. You can access that all with a free trial. I'll tell you a little bit more about that later. Now let's move on to the next phase, the link reaction and the Krebs cycle. The link reaction links glycolysis, this first phase of cellular respiration, to the next phase, the Krebs cycle. The link reaction unfolds in several key parts, and during the first part, pyruvic acid from glycolysis is transported across the outer and inner mitochondrial membranes, and it enters the mitochondrial matrix, this area over here. As that happens, enzymes remove carbon dioxide from pyruvic acid. They do that by snipping off this carboxyl group over here. That CO2 is 1/3 of the carbon dioxide that you exhale in every breath. In the next step, other enzymes will oxidize this remaining 2-carbon molecule. That's called an acetyl group. And that oxidation is paired with the reduction of NAD plus to NADH. That's the energetic output of the link reaction. And in step 3, enzymes will attach this 2-carbon molecule over here to a carrier molecule that's called coenzyme A. And that creates something that's called acetyl-CoA, acetyl coenzyme A. And that is the starting point for the Krebs cycle. The Krebs cycle completes the oxidation of glucose byproducts. What do I mean by that? This 2-carbon molecule over here that's being carried by acetyl-CoA still has a considerable amount of chemical energy. And all of that is going to be removed during the Krebs cycle. The oxidation of this acetyl group is going to power the production of 1 ATP, 1 FADH2, and 3 NADHs. You look at a diagram of the Krebs cycle and it looks terrifying, but I want to assure you that you only really have to know the big picture. The start of the Krebs cycle involves enzymes. All of these are enzymes over here. So the first enzyme will transfer this 2-carbon acetyl group from acetyl-CoA onto this 4-carbon oxalic acid. Note that it's a 1, 2, 3, 4-carbon molecule. That creates a 6-carbon molecule that's called citric acid. And citric acid, note that it has 3 carboxyl groups, and that creates a couple of alternative names for the Krebs cycle. It's called the Krebs cycle after its discoverer. It's called the citric acid cycle because of this compound over here, and it's also called the tricarboxylic acid cycle. In the middle of the Krebs cycle, enzymes will oxidize citric acid and its byproducts. Now, none of these byproducts are shown over here, but note that citric acid gets rearranged to another 6-carbon compound. We're not going to worry about its name. And then to a 5-carbon compound as the CO2 is removed. We're not going to worry about its name either. But note that these are all intermediate compounds within the Krebs cycle. So enzymes will oxidize citric acid and its byproducts. And as that happens, electrons and hydrogens from citric acid are used to reduce NAD plus to NADH. That's happening here. That's happening here, and it's happening here. They're also going to be used to reduce FAD to FADH2. That's that other electron carrier. At the same time, other enzymes are extracting chemical energy from citric acid and its byproducts and using it to do this over here at number 5, to power a substrate level phosphorylation of ADP and PI inorganic phosphate into ATP. I haven't used the term substrate level phosphorylation before, but it's very simple. In a substrate level phosphorylation, an enzyme, like this represented by 1, takes a phosphate and jams it onto ADP, creating ATP. That's substrate level phosphorylation. And it's called that because an enzyme is doing the work. That's distinguished from oxidative phosphorylation, which we're going to meet in a moment during the electron transport chain, in which oxygen acts as the final electron acceptor of the electron transport chain, and various cellular processes result in the formation of ATP. Here's the Krebs cycle and what you really need to know about it. The products are as follows. For each acetyl-CoA that enters the Krebs cycle, 1 ATP down over here, 1 FADH2 over here, and 3 NADHs over here, over here, and over here are generated. 2 CO2s are released as a waste product, and that's the other 2/3 of the CO2 that you exhale with every breath. Oxaloacetate is the starting and ending compound, and that is what you should memorize about the Krebs cycle. I have a song about the Krebs cycle, and I'm going to play you the last verse, which summarizes what I just taught.
We've walked through the cycle, so now let's review. Input is acetyl-CoA with carbons too. The carbons get removed, releasing CO2. Exhaling sends the CO2 out of you. The cycle's functions: energy transformation, 3 NADH1 FADH2 creation, and also synthesis of 1 ATP, which cells directly utilize for energy. For every glucose cells absorbed, the cycle runs 2 times. As long as cells get fuel, Krebs is running just fine. It precedes electron transport chain, it follows glycolysis. It's spinning round and round in the mitochondrial matrix. Krebs.
There's the Krebs cycle, and if you need more support about the link reaction or the Krebs cycle, you can find that support on learn-biology.com. With learn-biology.com, you will get the skills and the confidence that you need to ace your biology class and the AP Bio exam. Learn-biology.com has 100% alignment with the College Board's AP Bio standards, and it's been approved by the College Board as a textbook alternative. Hundreds of teachers around the country are using it right now because of that. Learn-biology.com gives you interactive tutorials with readings, videos, flashcards, and quizzes that teach you everything that you need to know about every topic in AP Biology. The interactions include multiple choice, click and drag diagrams, identify the part diagrams, and free response questions. You get positive feedback for correct responses, and you get hints and repetition for incorrect responses. Use learn-biology.com. You will wind up understanding the material so well. There are student learning guides that are available to you that tell students exactly what to do for each of our tutorials, and everything that you need to crush it on the AP Bio exam is provided to you in the form of FRQ reviews, review flashcards, multiple choice reviews, click on challenges, which are games, video resources to review for the AP Bio exam. Learn-Biology is the place that you want to go. There are student performance monitoring tools for teachers, a kind of grade book, so you can see how your students are doing. And free trials are available for both teachers and students. Don't delay. Go to Learn-Biology today so that you can crush it in AP Biology. Now let's end by talking about the electron transport chain. The electron transport chain, which is this last part over here represented by the stairway, oxidizes NADH and FADH2 that was created in the Krebs cycle, the link reaction, and the first part of cellular respiration glycolysis to power the phosphorylation of ADP to ATP. Most of the ATP created in cellular respiration is created during this last phase. How does this happen? We've created all of this NADH and FADH2, and that makes its way over to the inner mitochondrial membrane shown over here. And the first thing that happens is that NADH is oxidized to NAD+. The lost electrons then move into what's called the electron transport chain. The electron transport chain is a series of membrane-embedded proteins in the mitochondrial inner membrane. So this array of proteins, that's the electron transport chain. And you can think of it as a little wire that runs through the inner mitochondrial membrane. What this wire does is it powers proton pumps, shown at 3 over here. And what these proton pumps do is they pump protons from the matrix into the intermembrane space. And that creates an electrochemical gradient. Oxygen acts as the final electron acceptor. Oxygen's highly electronegative, so it pulls electrons along the chain. And when those electrons flow to oxygen, oxygen becomes reduced to water. As it does, it also absorbs protons from the matrix, and that increases the proton gradient between the matrix and the intermembrane space, with many more protons in the intermembrane space than in the matrix. Here's another way to think about this gradient. All of these protons are trapped over here. There are many fewer over here. That creates a lot of positive charge over here compared to the charge over here. Things always want to diffuse from where they're highly concentrated to where they're less concentrated. And that gradient is further powered by the fact that positive charges repel one another. So all these positively charged protons are pushing away from one another. You can think of those trapped protons as potential energy, like the water that's trapped behind a dam. And engineers build turbines to take that water in a controlled way and use it to generate electricity. That's hydroelectric power. Well, in cells, the trapped protons are used to generate ATP. And they do that by flowing through the only channel that allows those protons passage. And that's called ATP synthase. And its name tells you what it does. It converts ADP and inorganic phosphate into ATP. ATP synthase is a channel. It's an ATP synthesizing enzyme. And it's the only way for protons to leave the intermembrane space over here and return to the matrix. So what's happening is that facilitated diffusion of protons. The energy is already here in the gradient, and all these protons need to do is to diffuse through the ATP synthase channel. And as they do, their kinetic energy is used to power the formation of ADP and inorganic phosphate into ATP. So again, an exergonic reaction, which is the spontaneous flow of protons through this channel, is powering an endergonic reaction, the formation of ATP. Just to take another look at ATP synthase, here it is. You can see this channel for protons to diffuse through, and the structure of this molecule, it's really a complex of molecules, is quite remarkable because as these protons diffuse through, this entire structure will turn. There are binding sites for ADP and phosphate, and the turning of this structure over here will actually force the phosphate to bond with ADP, creating ATP. This is universal to life. Every domain, bacteria, eukarya, archaea, uses ATP synthase for the creation of ATP. The electron transport chain is also something that I've written a song about, and I'd like to play you the final verse leading into the chorus.
Now all these protons in the intermembrane space are trapped. They can't get out of that place, cause protons are charged and could never get through a phospholipid bilayer. They can't diffuse, but like all particles, they're dying to go from where the concentration's high to where it's low. Stuck in the intermembrane space, they're frustrated to diffuse to the matrix. They're highly motivated, and this gradient's been made steeper by O2, which absorbs protons from the matrix stew. So from proton pumping and oxygen's actions, add another force, electrochemical attraction. Think of all those trapped protons, each one's positive. The matrix in comparison is negative. Opposites attract, so the protons are dying to get to the matrix. Oh, how they're trying. There's only one channel that lets protons pass, and they use it like high school students busting out of class. It's a channel and an enzyme. It's ATP synthase, the closer in this game, an energy ace. ATP synthase is embedded in the inner membrane. How it works is so cool, it's insane. It's got channels for diffusing protons running right through it. When cells make ATP, well, watch how they do it. The matrix side of ATP synthase has binding sites for ADP and P, which come in and bind. And as ATP synthase lets protons barge through, their kinetic energy gets put to use. Like water through a turbine, proton movement generates rotation, changing synthases binding site conformation, which catalyzes chemical bond formation. ADP and P make ATP that energy sensation. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme, ATP synthase, which uses diffused protons' kinetic energy to make ATP from ADP and P.
That's the electron transport chain song. That sets us up for our weekly quiz. We're on the Unit 3 review page on learn-biology.com. There's an overview video. There's a list of objectives. There are flashcards, but this is what we're looking for, the Unit 3 multiple choice quiz. We want to test about cellular energy and cellular respiration. That's a total of 9 questions. Okay. Which of the following must be true of the coupled reactions shown below? The choices are as follows. Choice 1, reactions 1 and 3 are exergonic. Reactions 2 and 4 are endergonic. Choice 2, reactions 1 and 2 are exergonic. Reactions 3 and 4 are exergonic. Choice 3, reactions 1 and 3 are endergonic. Reactions 2 and 4 are exergonic. And the fourth choice is reactions 1 and 2 are endergonic. Reactions 3 and 4 are exergonic. I'm thinking that the reactions where the reactant has a higher amount of energy than the product must be the exergonic one. That's true of 1. That's true of 3. 2 and 4, it's the opposite. Let's look at choice 1. Excellent. In both reactions 1 and 3, the reactants, glucose and ATP, have more energy than the products. That indicates that these reactions have to be exergonic energy releasing. Reactions 2 and 4 are the opposite, indicating they have to be endergonic energy requiring. Let's move on to another question. If cultured muscle tissue is kept under aerobic conditions and provided with 10 moles of glucose, what is the maximum number of moles of ATP that this tissue will be able to produce? For every molecule of glucose that comes into cellular respiration, the cell can create 2 ATPs in glycolysis and then more ATPs, 30 plus, in the electron transport chain. So as I look at these choices, we have 10 moles of glucose. What's the maximum number of moles of ATP that this tissue will be able to produce? By far, the closest is this 360 over here. Let's click on it. Nice. Under aerobic conditions, 1 mole of glucose can power the production of up to 36 moles of ATP. Here's a question about the uncoupling channel. The uncoupling protein, UCP, is a protein channel that can be found in the inner mitochondrial membrane, as shown in the image below. The protein is expressed in chipmunks and other mammals during the winter. What would be the effect of expressing this protein in terms of ATP production? We have a diagram of the uncoupling channel, and it shows protons diffusing through the uncoupling channel instead of diffusing through ATP synthase. So the choices are: more ATP would be produced because UCP allows more protons to flow through the ATP synthase channel. Choice 2, less ATP would be produced because UCP allows fewer protons to flow through the ATP synthase channel. Choice 3, more ATP would be produced because UCP pumps protons up their concentration gradient. Choice 4, less ATP would be produced because UCP pumps protons up their concentration gradient. I'm going to deliberately choose a wrong response. I chose less ATP would be produced because UCP pumps protons up their concentration gradient. And the feedback that you're getting is no, the UCP channel is not a proton pump. It's a channel that allows protons to bypass the ATP synthase channel. That's the kind of hint you get in response to wrong responses on learn-biology.com. We might see this question again, depending on how many questions we do. Next question. Researchers conducted an experiment to test the efficiency of ATP production in white-tailed hornets' wings muscles. They measured ATP production at various flight speeds in a wind tunnel. At the highest speeds, the hornets were exhausted quickly. The results of the experiment are displayed in the diagram below. The diagram is a very simple diagram. The x-axis is flight speed, the y-axis is ATP per glucose, and it shows that at lower flight speeds, the ATP for glucose is high, and then it dramatically falls. And at the highest flight speed, it is much, much lower. So, which of the following statements best describes the pattern in this diagram? 1. Glucose is reduced at low flight speeds and oxidized at high flight speeds. 2. Glucose is the main energy source at low flight speeds, but more energetic molecules are used at high flight speeds. Choice 3. Oxygen is the final electron acceptor at low flight speeds, and fermentation is used at high flight speeds. 4. Electron transport is used at low flight speeds, but slows down metabolism at high flight speeds. When cells that are in aerobic organisms can respire aerobically, meaning the entire process of glycolysis, link reaction, Krebs cycle, and electron transport chain, they'll do that and produce 36 molecules of ATP per molecule of glucose. When there's insufficient oxygen, organisms like us will switch over to anaerobic respiration. They'll just do glycolysis followed by a fermentation reaction. And that actually is something we're going to talk about at the very end of the show. So hang on until then. The answer is the third one. Oxygen is the final electron acceptor at low flight speeds, and fermentation is used at high flight speeds. The explanation: at low flight speeds, the muscles in the hornet are using the entire aerobic respiration pathway, glycolysis, followed by the link reaction, followed by the Krebs cycle, followed by oxidative phosphorylation. This pathway creates over 30 ATPs per glucose. At the highest flight speeds, the muscles are exceeding the capacity of the hornet's respiratory system to deliver enough oxygen. As a result, the hornet switches to anaerobic respiration, which yields only 2 ATP per glucose. Next question. During cellular respiration, what is the function of the mitochondrion? The first choice is hydrolyzing ATP. The second is oxidizing water to oxygen. The third choice is oxidizing pyruvate completely. The fourth choice is converting glucose into pyruvate. The fifth choice is generating NADP plus from NADPH. A good strategy in multiple choice questions is to use a process of elimination. Hydrolyzing ATP. No, that's what cells do when they do work. That's not what's happening in the mitochondria. Oxidizing water to oxygen. That happens in photosynthesis, which we'll talk about next week. Oxidizing pyruvate completely. Not how we usually think of cellular respiration, but that's an interesting choice. Let's look at the other two. Converting glucose into pyruvate. No, that happens during glycolysis, which is in the cytoplasm. Generating NADP plus from NADPH. No, that's a process that happens in photosynthesis. It's choice number 3. Next question. The graphs below show the energy changes in 2 reactions. Which of the following statements is correct about these reactions? In diagram 1, the reactants have more energy than the products. In diagram 2, the reactants have less energy than the products. So here are your choices. Reaction 1 is exergonic and shows energy changes in a process like photosynthesis. Choice 2. Reaction 1 is exergonic, and C which is the energy difference, represents the energy required to convert the reactants into products. Choice 3. Reaction 2 is endergonic and could show the energy profile of a process like photosynthesis. 4. Reaction 2 is endergonic, and D shows the energy difference between the products and the reactants. The answer to this question is the third choice. Reaction 2 is endergonic and could represent a process like photosynthesis. That's because photosynthesis takes low energy reactants, carbon dioxide and water, and converts them into high energy products like glucose. In the series of reactions below, which letter represents an oxidation reduction reaction? The diagram that we're looking at is glycolysis. A shows ATP being added onto glucose. B shows fructose 1,6-bisphosphate being split into two G3Ps. C shows an enzyme converting NAD plus into NADH. D shows the cell oxidizing or using the chemical energy in G3P to generate two ATPs. So which of those, A, B, C, or D, represents an oxidation reduction reaction? It's the one where NAD plus is being converted to NADH. That's choice C. Great job. In stage C, G3P, or one of its byproducts, is oxidized so that NAD plus can be reduced to NADH. Let's keep going. The diagram below compares energy metabolism in normal cells to cancer cells. LDHE stands for lactose dehydrogenase A, an enzyme that converts pyruvate to lactic acid. MCT is a channel that allows lactic acid to diffuse across the cell membrane. There's a diagram that's comparing a normal cell to a cancer cell. And the main difference is that in the cancer cell, there's an arrow from pyruvate that goes to lactate and then goes into this channel called MCT. Whereas in the normal cell, from pyruvate, there's an arrow that goes into the mitochondrion that leads to oxidative phosphorylation. Based on the diagram, which of the statements is most likely correct? First choice, cancer cells perform more glycolysis than normal cells and are less efficient at generating ATP. B, or the second choice, cancer cells perform less glycolysis than normal cells and are more efficient at generating ATP. Third choice, cancer cells perform more glycolysis than normal cells and are more efficient at generating ATP. And the fourth choice, cancer cells consume less glucose than normal cells and are less efficient at generating ATP. I'm looking at the diagram of cancer cells. I'm seeing that very little pyruvate is going into the mitochondrion. I'm assuming that the result of that is that cancer cells would perform more glycolysis and they'd be less efficient at generating ATP. And that's correct. If you compare the cancer cell to the normal cell, you can see that in the cancer cell, the arrow between pyruvate and lactate is thicker, indicating that more fermentation is occurring. In addition, in the cancer cell, the arrow between pyruvate and the mitochondrion is thinner, meaning that less pyruvate is going into the mitochondrion to power oxidative phosphorylation. Together, that indicates that more glycolysis is occurring and glycolysis is less efficient at generating ATP. That's the kind of practice you get on learn-biology.com. Now let's talk about your plan for this week. You should go to learn-biology.com, then the courses menu, then AP Bio, and then Unit 3 Cellular Respiration. We've got tutorials about everything that we've talked about. If you invest a little bit of time, 10 minutes a night, you will make huge gains because you'll be interacting with the material. There is one incredibly important topic that we did not cover in this video. It came up a couple of times in the quiz, and that topic is anaerobic respiration and fermentation. Incredibly important to understand what cells do when oxygen isn't present. You can learn about that in the next video, which I'm posting right here. So watch the next video. Please subscribe to learn-biology.com. Subscribe to the Science Music Videos channel. Watch the next video. I'll see you next week. Thank you so much.